So workshop 4 is all going to be about experimental design. So this is really going to underpin a lot of what you learn about in your degree. You're going to be encountering lots and lots of different experiments, both in terms of things that you see in the lectures, but also in terms of experiments that you do yourself. So in a couple of weeks' time, we're going to be in the lab, we're going to be doing some experiments. So having a really good sense of what makes a good, robust experiment is going to be really important for you as you go through your degree. So, as you know from the skills module, when we want to investigate something, what we usually do in biology is to set up a hypothesis. So this is our research question, this is the thing that we're trying to find out. So in this case, my hypothesis is that we've got a new drug, let's say it's a new drug that a pharmaceutical company has developed. We're going to ask the question, does this drug increase the rate of apoptosis in HeLa cells? So apoptosis is programmed cell death. HeLa cells are those human cancer cells we learned about a couple of weeks ago. So what we would obviously like to do is to treat some cells with our drug and measure how many of them undergo apoptosis. Okay, that's a fairly straightforward thing. We'd like to know how many of them die in the presence of the drug. For the purposes of this, what I've given you, let's assume that drug X is a drug that's not directly soluble in water. So what you have to do, just because of the chemical properties of drug X, is that you actually have to make it up in a dilute solution of methanol. It won't go into water straight away. And that's true of an awful lot of pharmaceutical drugs. They're not water soluble. You have to add a little bit of methanol in this case in order for them to work. Okay, so there's our measurement. So we've treated the cells with a drug, we've measured how many undergo apoptosis, but that's not a particularly robust experiment. There's lots of things that we could do to improve this as a research design. So first of all, just to check that you're happy with some conventions. So usually in an experiment, we would have our independent variable. So this is the thing that we are changing in the experiment. We usually have that on the x-axis. So in this case, this is the treatment that they are receiving, and then we usually have our dependent variable or our response variable on the y-axis. So in this case, this is the percentage of cells undergoing apoptosis. So by convention, we have our variables that way round. Okay. But this still isn't really an experiment because all we've done is to give them the drug. We have nothing to compare it to. So we obviously need to have some sort of control in there. And one of the things we need to have is a negative control. So one negative control would be to have some untreated cells. Okay, so we have some other cells that have not seen the drug. We also measure how many of them undergo apoptosis, and then we can start to compare what's happened with the two of them. So that's a better experiment. But as you know, then making a single measurement isn't really very good experimental practice because you don't know how representative that measurement might be. So what we need to do to improve this is to add some replicates. Okay, so we might have to do this experiment 10 times for each treatment. So those are our replicates, and then that allows us not to calculate a mean and a standard deviation, or to make the box plots like you learnt in the statistics module. Okay, so that's now starting to be a respectable experiment. We've got a negative control with a number of replicates. We've got our treatment with a number of replicates, so we can start to make some comparisons. But there are still things we could do to improve this experimental design. So one of the things here is we've actually added two things in here compared to our untreated cell. We've added the drug, but we've also added the methanol because of the chemistry of the molecule. So another thing that we probably ought to do is to include what in this case would be a solvent control. So we need to say, well, what happens if I just add the equivalent amount of methanol? Okay, so if your treatment comes in combination with anything else, then you need to be able to design it so you can separate those two out. So the way we would do that here is we would give them just 1% methanol without the drug. We would compare it to the untreated, so hopefully there should be not that much difference. But then when we add the drug, then we can see the effects separating out. So particularly for biochemistry, solvent controls can be really important. Another thing we might want to do is to compare it to another drug that we already know works. And this would be an example of a positive control. Okay? So here we've got a known drug, so maybe this is one that's already on the market or something we know from some other experiments would work. That's a positive control, so we know that that does the job, so we can then have sort of better consistency within our experiment, and then we can compare the new drug with the known drug. So that will be a positive control, something that we know will respond to the treatment. And then particularly when we're dealing with biochemistry or pharmacology, one of the things that we often do is not to just give one concentration of the drug, but we actually might give a range of different concentrations because we don't always know what the effective biological concentration would be. So here we've got different concentrations of the drug. So it may be that low concentrations, so 1 micromolar here, the drug doesn't really do anything, but at high concentrations it's much more effective. So those are all sort of characteristics of good, well-designed laboratory-based experiments. You need to have, you might have multiple different controls. You might have negative controls, which aren't going to do anything, positive controls, which you know should do something, and then you've got various other controls in there, and obviously all of them measuring a number of replicates so that you can then do statistics on them. So we're going to be thinking about these sorts of experimental designs. So what we're going to do in the workshop is to actually start to read a scientific paper. So I've uploaded one on Canvas for you that we're going to have a look at in the workshop to both think about the experimental design, but also to think about some strategies for attacking reading scientific papers, because it's not always very easy. So just to be aware of the structure of scientific papers, we usually have an abstract, which is a short summary of the whole paper. So that will give an overview of what the whole paper is about. We then have an introduction, which is the scientific background to the study, and will state the aims or the hypothesis somewhere in there, so we can say what the study's all about. We will then have the methods, which is the actual protocols that are used. Now, in the paper I'm giving you, the methods are actually in supplemental information. They're not in the main bit of the paper, but they are presented there. So just look to the end of the paper to find them. We've then got the results, which are the figures and the tables, but also a description of the findings. So we'll look at how those sections are written. And then discussion sections is where the authors interpret their results. They maybe criticise anything that may be wrong with the experiments, and they discuss the broader context of the results, so why that might be important for our understanding of biology in general. So we're going to be thinking about how to approach reading a paper and what features of experimental design we might think of in designing a good study. Now, a word of warning, reading papers is quite challenging. It's quite difficult the first few times you do it. I've uploaded a PDF onto Canvas of how to read a scientific paper. It's a really nice guide written by the American Society for Plant Biology, but it's the same for all scientific papers. And we'll discuss some hints and tips of how to read papers effectively in the workshop.